The next part of our activity content this week is enzymes. So when we think about enzymes, enzymes are basically small molecules. They usually end in the word ASE. So when we see them, or ACE, we can identify them right away. And their job is basically to work with our metabolic reactions, and they are going to speed up the reaction. They're going to lower the energy needed for the reaction to occur. They usually need another kind of helper molecule that we call a substrate. And they usually, when they match up with that substrate, create a new product that basically is made from the substrate so that the enzyme is a recyclable molecule. It doesn't really get used up in any way. So reactants are reluctant to participate in the reaction, so energy must be added at least one reaction to initiate things. So sometimes we do need enzymes, but we also need to have a little bit of energy to help them along. Most times we don't need energy for enzyme work. So this is just a graphic that shows enzyme and energy activation. So the energy of reactants is here. As we move up the chart, this is what we call free energy, and this is the progress of their actual reaction. So you can actually see with these two lines here that kind of go up and then end up in the same place, this is activation energy with an enzyme, so this area right here. So this is with energy and an enzyme, but this is with activation energy without an enzyme. So we use a lot more energy, because remember, the higher we go up here, the more energy we are using. So if we have to use kind of the activation energy and we have an enzyme that will help, we use less overall free energy to get to the same exact product, okay? So just be aware of that. We can see different structures. Now, enzymes are structured molecularly very specific. They have a specific shape, and within that shape we have what we call an active site. So you'll see, like, this is kind of a semicircular area, and then it has like these two kind of semicircles up top. That's a specific shape. This enzyme has this other very unique shape. So each enzyme is very specific, and it can only work with certain substrates that fit with it. So they have to kind of be like a lock and a key that has to fit exactly into the lock can only make it turn and open. Within this enzyme, we have this active site, and that's the site where the substrate needs to fit. So for in this example, this red substrate needs to fit here, and the green substrate needs to fit here, and then we would have a complete structure. Here, these shapes are a little different, and again, they would fit very nicely in the specific shape of that particular enzyme. So here's another view of it. The enzyme here is kind of blue, looks like an E shape a little bit. The substrate that works with it has this other unique shape. You can see that it fits very nicely into what we call the active site. When they kind of lock and key, or what we call induced fit, together, they become a new unit, and this is the enzyme substrate complex when they kind of fit together, enzyme and its special substrate. When we move over here, what happens is that we have the enzyme, which is exactly the same. It's shaped the same as when it did in the beginning because it's unchanged, it's unused. What changed? The substrate did. In this case, it turned into two new products. Sometimes it might just turn into one product. Sometimes it could turn into three products. It depends on the enzyme and the substrate. But the enzyme is the same. The products are going to go along its way and do what they need to do. The enzyme is now available for another substrate to come in. So it has the ability to definitely be recycled, reused. So here's that enzyme specificity. We can have two substrates that come in, like I showed you an example earlier with the red and the green circle. Here's our enzyme substrate complex, and in this example, they release as one new larger product. So again, it could be smaller products, it could be a larger product, it could be multiple products, but the product is the new change. The enzyme stayed the same. Even with this example that I just showed you, enzyme looked the same every single time. It does not change. So that's what we call, I said lock and key a moment ago, but induced fit model is another way to talk about it. The enzyme substrate complex where the enzyme changes, this does not change its shape. It kind of like takes in the substrate, holds on to it, so it kind of grabs onto it like a handshake, right? And then once it's ready to release its new product, it goes back to that normal shape. So it holds on to whatever comes into it that fits. So here's a visual. Here's our enzyme, here's our substrate. substrate, it's going to enter the active site. And so what happens is the enzyme kind of squeezes it, clamps down around the substrate, forming that induced fit. So they fit together like a glove on a hand. And it holds it there because it doesn't want it to leave. What's going to happen is chemically that substrate is going to change within here, and when it's ready to be released, the enzyme opens back up and it will just be exactly the same structure as it previously was. Now, there are a few things that might affect how the enzyme works. So that could be temperature, it could be pH. So let's talk about those. Temperature. If we are in extreme heat, very high heat, really hot, or very cold heat, the enzymes are not going to work. And what happens is that we call denaturize. So they denature, they break down. Extreme heats, extreme cold, so temperature change, permanently change the shape of the enzyme so it can no longer work. So optimally, our internal body temperature is 98.6-ish Fahrenheit, about 37 Celsius, and there's a small average range for folks. That's the temperature these enzymes need to work properly in. Okay, the other factor that might change how it works is the pH. So we want to have optimal pH. Now, pH of blood is about 7.4, but the pH of your stomach should almost be a 1 or 2. It is very acidic. So there are specialized enzymes that might work in that environment, but not all enzymes can. So they need to be at an optimal pH level in order for them to work, or again, they're going to denature and change shape and not work. Now, substrate concentration basically means if there are more substrate around, obviously there's more activity, right? Because the enzymes are not changing, they're staying the same, they are available for new substrate. So if the substrate is like very high, we can turn over new products often. But again, there's a point that we can do that at because it does depend on how many enzymes are around. Now, there are a few things that we do because sometimes enzymes work so great in our body that they work to a fault, meaning that they're working too much. So sometimes we have things that don't work enough. Lots of times enzymes can work too much. And so when that's the case, we need to have an inhibitor, an inhibition, a kind of stopping of that. So we can have some different types. We can have obviously an irreversible, which is basically kind of a permanent poison that changes the enzyme. And we don't see that a lot, but again, think about it, it's a poison. Now, reversible are more common, but we can have two types. So we can have what we call a competitive, inhibitor, or we can have a non-competitive inhibitor. So a competitive molecule basically competes to get into the active site before the substrate. And when it does, it shuts down the enzyme. And a non-competitive, kind of reversible one, molecules bind to another site kind of on our enzyme, and it turns off the active site, causing the enzyme to kind of change shape. So let's look at a picture here. Here is an example of a normal reaction, right? So this is our little kind of almost like a Pac-Man, and then we have a substrate that kind of has a diamond shape with a circle. Here's the active site within the enzyme. So normally, like it could, this little diamond fits right into the mouth of the little Pac-Man enzyme, and that's your enzyme substrate complex. It goes through the reaction process and produces two new products. So we have a diamond product and we have another little Pac-Man little face, okay? That's our normal reaction. But an inhibition is this. Basically, an enzyme is here. It has this very same kind of shape to it that fits into the active site, but the other end has an inhibitor. It's a different changed type of molecule. So what happens is this comes in, fits in nicely. Here's our active complex, right? But what happens is our normal substrate comes along and it can't fit in there because this is already being used. And remember, when this comes in, the enzyme kind of clamps down on it like a handshake and holds onto it. Problem is it never releases because there's no reaction. So this is basically an inhibitor. It is binding to. This is an example primarily of competitive inhibition because this is a competitor, looks just like the active site would want it to look like, the substrate would look like, and it gets there first. So it's in competition. Here's a non-competitor. So this is exactly a similar example. Here, our enzyme actually has an inhibitor site. So some of our enzymes do actually have that. In this case, here our substrate comes in, all right, binds, same process. 2 new pieces, 2 new products. But what happens when we have the inhibition in place? Again, here's the inhibitor that fits right into the inhibition site. And when it did that, look at this, it changed the shape. So when it changed the shape, yes, the shape still kind of had this active site piece, but our substrate bonds to it. And when it bonds to it, it can't do anything. It basically can adhere, but there's no reaction that's going to occur. So we deal with inhibitors a lot in the medical field. So that again, things can work more efficiently, not as competitive as it normally would, because we do have enzymes that work too hard sometimes, and they're overworked, and they're doing the body harm because they're doing too much work. So again, competitive versus non-competitive and enzymes. We're going to have a whole entire separate lab on enzymes when we get there after the next few weeks. So you'll hear more about enzymes at some point in the next few weeks.